believe most of you probably talked quite a few times during the recruitment process, right? Okay, so you should be asking. You should be asking for career advice here. Yeah. Okay, so conspiracy theories and deep state aside, I will hand over.
Thank you so much. All right. Well, it's really a pleasure to be here today. Thank you for giving me the opportunity to speak to the DPRG. And it's funny because I actually gave grand rounds this talk actually last week to the Department of Endocrinology. And people said, oh, nobody in the department has actually really ever heard you give a talk. I mean, not really. I mean, you've heard me make very brief presentations, but not really to kind of give you an idea of the kind of research that I've been conducting over the past, this is probably more along the lines of the past 7 years or so. Tracy Bale and I, when we were at Penn together, she's now at the University of Maryland, sort of had a marriage made in heaven. She is a basic scientist, and myself as a clinical researcher, we're able to really work together through the use of a P50 mechanism, which, for those of you who know, the program sort of content, sort of program project kind of mechanisms. Why is there noise on the other end? I think people who are Zooming in, maybe you want to mute yourself. Thank you. Okay. I don't need any more competition than I already have. But in any case, it's really, an amazing opportunity as a clinical researcher to be able to work with a basic scientist and to be thinking more mechanistically and in a translational way in the research that we're doing. So I'm going to highlight a little bit of that in this talk. But moreover, I really just want to tell a story about why do we need to be thinking about childhood adversity, early life stress when it comes to health outcomes decades later? And what about sort of reproductive transitions when particularly females are undergoing sort of dramatic hormonal fluctuations? Why do we need to be thinking about, early life stress interacting to show risk or resilience during those particular time points in an individual's life? So, and we have, what, an hour for this? Okay. So I'll try to keep it shorter than that, if at all possible. I do have a few disclosures. One that I highlight is with Sage Therapeutics. How many of you heard about brexanolone, which is the new antidepressant for postpartum depression? I ran the phase 2 and phase 3 clinical trials at Penn as the site investigator there, and have done a number of consultations and education seminars for Sage, specifically about postpartum depression. And yes, I was paid for my consultative time. So just today, we're going to talk about the importance, obviously, of a developmental lifespan approach. I don't have to really belabor this point for you guys, given you are the DPRG and developmental psychobiology is what you're all about. But I also want to talk a little bit about the interactive and individual effects of stress and gonadal steroids. And I could spend a whole lecture just talking about the impact of hormones on neurotransmitter systems, neuronal structure, glial cell morphology, how closely the glial cell is interdigitated to the synapse and sucking up glutamate, and how estrogen modulates. I mean, there's a whole host of things that we could talk about when it comes to gonadal steroid effects on the brain. Also, stress effects on the brain. Obviously, those are separate lectures and could take, a whole course to get through. But I want to use the estrogen, serotonin, and stress interaction as just sort of a model system, mainly because it is sort of a really good example of basic science research and clinical research implicating this sort of interaction, particularly with serotonin as the neurotransmitter in question, but also because the protocol that I use in my menopause population to look at the effects of estradiol on the brain and how estradiol may be mediating some of those effects through different neurotransmitters, I use a serotonergic manipulation. So that's one of the reasons that I focus on this one in particular. Not by a long shot is it the only neurotransmitter hormone interaction we could discuss. And then obviously you all know about the enduring impact of childhood adversity, but we're going to talk about it in the context of pregnancy as well as the peri and postmenopause. So I know that this is not something that is new to any of you, that the first 9 months of gestation is your first environment, and that what happens during this time, particularly for mom, can have a dramatic impact on the risk and resilience for the offspring, whether it be medical. Well, I don't like to separate psychiatric from medical. It's all the body. Whether it's a brain organ system that is affected, or cardiovascular, or metabolic, or immune, we all know that there are things that are going on for mom, not only psychosocial factors that could contribute to risk or resilience for her pregnancy or the outcome of the offspring, but also it could be, her nutritional status, how well her placenta is functioning. As you probably know, the placenta is primarily from fetal origin. As a pregnant woman, you have very little, input on how that placenta functions. And is that placenta actually, functioning in a way that is deactivating cortisol, for example, the way it should through 11 beta HSD activity? Does that placenta pour out more corticotropin releasing hormone than somebody else's placenta? Again, all of these factors can contribute to how well the baby develops, but also not just about the baby, but the health of the pregnancy itself. So we know that timing of gestation can be impacted by a number of these variables as well. And so again, this intrauterine programming is becoming something that most people are thinking about. But the one thing that I also want to point out is that it's not just about what happens during pregnancy, but there's plenty of evidence from animal studies as well as human studies now that what happened to mom earlier in life programs her own HPA axis response across her entire lifespan that then interacts with these pregnancy changes in the HPA axis. And so you've got this unique hormonal milieu that is occurring during pregnancy that could be impacted by mom's early life experience. Now, there's also data that shows that father's life experience, now whether it's childhood adversity or it's more proximal to the time of conception, but because of some of the work that I've been able to do with Tracy, we've actually got a paper now in front of Nature Neuroscience focusing on acute stress for the male. So a male who is stressed right now, what are the changes in their sperm in the next 3 months? And we've actually been able to see that there are characteristic changes in things like microRNA and various vesicles that you find in sperm and semen, and how, just even reprogramming glucocorticoid receptors in the epididymis can have an impact on those sperm that then have in animal studies behavioral effects. And so we have a lot to learn. We focus a lot on moms and thinking that moms are responsible for intergenerational transmission of information to the offspring, but it also is important in dads, and we have a lot more to learn in that respect. So again, this is a very interesting area to work in, and I'm really lucky that I have a collaboration with a basic scientist so that we can get more mechanistic in our understanding of these issues. So basically, I don't want to make it sound like it's all about mom and dad. It's sometimes about the baby as well. And so how many of you were born into this world with this as your temperament? Seriously. I mean, it's great if you're like sunny and gay and happy and you're just like able to accept everything that comes your way and it kind of stress kind of rolls off your back. But I would imagine, since we're all in academia, that this is really how we came into the world. You have to be a little tough, maybe a little paranoid and a little suspicious in order to be able to make it an academic world. And so, but again, this is how your offspring also interacts with the world. And so it's not all about mom and dad. And a lot of times we as parents, I think, take on too much responsibility for thinking that, we created this child, and if the child has a difficult time, it must be our fault. Your child is a separate individual. I hope we all recognize that. And we have very little control over who we get and how that infant comes into the world. But it is a complex interaction between mom, dad, what's happening prior to conception in utero, and then how the baby sort of interfaces with the experiences that it's going to have. And ideally, there's a match between mom, dad, and in utero programming and the life that the baby is going, or the environment the baby is going to be exposed to afterwards. But a lot of times there's a mismatch. And obviously that can also be quite problematic as well. So we know that there are these critical windows across development where stress or adversity or just, life hassles, what have you, lack of social support, whatever it might be, that, these stressors are going to have a different impact on the system. And when I say system, I mean things like HPA axis function or, cytokine response to stress or, again, basic physiologic responses to the environment can be altered depending upon when these events occur. Obviously, if it's in childhood, when, the amygdala and hippocampus, earlier in childhood are sort of really developing and those cells are, maturing and you're making pathways and connections, an insult at that time is more likely to obviously have an impact on a system that is basically rapidly developing. And that would be when you would expect to have more negative effects on those particular systems. If you're, again, somebody who is having a trauma or a difficult event or lots of difficult events that you're trying to deal with as an adolescent, maybe prefrontal cortex function would be more effective because that is really part of the brain that is developing most rapidly during that time. But we also know that even after the brain is fully developed and you're not sort of having sort of these adverse events occurring on top of a developing central nervous system, we also know obviously that stress and trauma later in life can lead to PTSD and depression and other factors. And even, for example, Janice Keycolt-Glazer's work looking at caretaker stress. For example, if you're an elderly person and you're caring for a partner that maybe has Alzheimer's disease or some other medical condition, she has shown that even stress during that time and somebody who's not had adverse life experiences, but that particular stressor has an impact on, for example, their ability to fight off infection. So changes in their immune response. So we need to be thinking about stressors across the lifespan. And some of the data that we have suggests that, there might be a two-hit model, that you undergo early life stress, but then it's the stressors later in life that kind of unmask the risk. And again, one would arguably say that maybe that person is very resilient until they undergo a second stressor. Some people, it's, whatever it is, one stressor is enough. They don't need two hits in order to experience the adversity. And then obviously we know that even if you're stressed, when you're, all the animal studies show that if you have that social support of the mother or your litter mates, that you actually dampen the negative effects of the stress. And that actually stress in the context of social support actually seems to lead to resilience. And so a lot of people always ask, well, in your studies, how much do you assess resilience? And I'll get to that because I think assessing resilience, and maybe we can have a discussion among some of you who do really focus on resilience, how you assess it, because it just to me has been elusive. Is resilience lack of disease? Is resilience the fact that you underwent a lot of stressors and you were able to get through a college education and have a meaningful relationship and you're married and now have children? So what if you suffer from depression? As long as you're responding to treatment, that's maybe really resilient. So again, what is it that we use to define resilience? And that's something that I just don't have a good answer for. All right, so basically, in my lab when I was at Yale, I started out as a women's mental health person. I didn't think about sex and gender differences per se. I just was focusing on, these sort of conditions that women were coming to me with, particularly in the postpartum period or during pregnancy. I then got very interested in premenstrual dysphoric disorder and got NIH funding. My first K was actually on PMDD. So, again, I started thinking about sort of hormonal changes across the lifespan, mainly because, one, I was treating patients as a resident and trying to understand how is it that some people come to me and they seem to have a difficult time or have an unmasking of a psychiatric problem, or at least suboptimal mood symptoms or suboptimal ways of thinking about the offspring. Why is this happening to them and not every other pregnant or postpartum woman? Why is it that only 5% of women have premenstrual dysphoric disorder when basically all the studies at that time in the 90s and early 2000s, people were looking at blood levels and saying, oh, well, they have more or less, estradiol or no. No, the bottom line is that most of the studies of postpartum depression, perimenopause depression, depression or mood symptoms related to menstruation, all the peripheral hormone levels were like completely not informative. I mean, they basically had very similar hormone levels if they had symptoms or if they didn't have symptoms. And, again, you could say, well, this is no brainer now, but back in the 1990s, people still thought that, oh, you could predict who's going to have postpartum depression just by looking at hormone levels. And it was pretty much in the 90s with Dave Rubinow and Peter Schmidt's work from the NIMH. They basically said, no, these women are just sensitive to these hormonal changes. There's something going on in the central nervous system that even if they have normal fluctuations in these hormones, this is not the reason. The fluctuations are not the reason that they're not abnormal fluctuations. They're normal fluctuations. It's just the brain's not responding in an optimal fashion. And so we have to look at central nervous system markers, not these peripheral markers of hormone levels. And so in any case, that led people to doing a lot more brain imaging and trying to understand, at least in my lab, trying to understand what can we learn in perinatal women that might inform us about menopausal women, or what we know about puberty and the onset of these fluctuations at puberty that might inform us to what's happening in the perimenopause, for example. So again, thinking about how hormonal changes that you study in one part of the lifespan might help inform us as to these changes out at later times. Because one of the things that we know about menopausal depression is that you're at increased risk if you have a history of severe PMS. There are a lot of women who I would see in the postpartum period who had never had premenstrual dysphoric disorder, and then all of a sudden after having a baby and they started menstruating again, they would start having a tremendous PMDD, and you would be like, wow, like what happened in pregnancy to trigger this for this individual? So again, it brought people's thoughts back to this possibility of this kind of kindling effect of hormones. So in any case, we know that during puberty, when you have onset of menstruation, that's when you start to see the sex bias in females for affective disorders, depression, different anxiety disorders, risk for PTSD. Then in pregnancy, obviously, you start to see sort of a perinatal mood symptoms. Again, postpartum depression being something that occurs in about 10% of postpartum women. And then in the perimenopause, we know that you're at increased risk for depression, even if you've never had a depressive episode. So you might be a woman who's never had a depressive episode. You're still 2 to 3 times more likely to have your first major depressive episode in the perimenopause. So something for all of you young ladies to look forward to. Having been through it already, I'm glad that's done. So the bottom line is that we know that these hormones are sort of, especially in females, really of interest when it comes to thinking about affective disturbance. Not in everybody. And one of the things that I've always done is tried to understand the healthy control, so to speak. How do some women go through these massive hormonal manipulations and not get depressed? I mean, what is it about them that is not changing versus the people that do have these really severe mood symptoms? Now, the other thing that's very interesting is that there are a lot of other medical conditions that do fluctuate with menstruation or pregnancy or the perimenopause. Migraines are very sensitive to hormonal changes in some people. Seizures, epilepsy, there's catamenial epilepsy, people who will have a clustering of seizures in the perimenstruum. So during the time that the hormone levels are declining and staying low in that early follicular phase. And so again, GI conditions, autoimmune disorders. So again, some people are very sensitive to hormonal fluctuations, and that seems to be consistent across the lifespan for females. Now, males have a less of a dramatic alteration in hormone levels across the lifespan. And, again, it just basically means that they don't have to respond to these internal perturbations quite as much. It doesn't mean that men don't have their own risk factors for psychiatric conditions. If you think about the disorders that are more common in males than females, or for which men have a worse course. So even though schizophrenia risk, may be a little bit higher in males than females, but we do know that when it comes to outcomes, response to medication, age of onset, that there are these sort of sex differences. But the disorders disorders which are really very clearly more common in males than females occur in males and are usually diagnosed in males before they undergo puberty. So for them, it seems like that, again, we talk about those being more neurodevelopmental. Again, I don't need to give you a lecture on neurodevelopmental disorders given you're the DPRG, but the bottom line is that those disorders seem to be more linked to either what's happening in utero and with early development, or perhaps, again, how much are androgens playing a role in those particular disorders, but in utero exposure to androgens and estrogens and not the sort of post-pubertal exposures. And so, again, when I'm thinking about sex differences, I do think about sort of not only what's happening with these hormonal fluctuations, but also what happened in utero to increase this risk for males of these neurodevelopmental conditions like ADHD, autism, and what have you. So as I promised, I won't give a whole lecture on how hormones affect the brain, because again, that would be a course. And, maybe we should have a course like that, but I'll do that in my spare time when I have spare time. The bottom line is that estradiol and serotonin have very interesting relationship, and particularly when it comes to early life stress. So I'm just going to kind of take you through only some of the pro-serotonergic effects of estradiol. So estradiol increases messenger RNA expression for tryptophan hydroxylase. Tryptophan hydroxylase is the rate-limiting enzyme, as you know, for conversion of tryptophan to serotonin. So if you have more estradiol on board, you have more TPH2 gene expression. You also, once you release serotonin into the synaptic cleft, you have decreased degradation of serotonin by monoamine oxidase. So basically, estradiol is going to decrease the activity of this enzyme so that the serotonin stays in the synaptic cleft a little bit longer. Now, what happens then? Well, interestingly enough, you actually get an increase in postsynaptic 5-HT2A receptor density. And we were able to show that even in menopausal, postmenopausal women who have been hypogonadal, using PET imaging with a ligand to the 5-HT2A receptor, we were able to show that even in a postmenopausal brain, so somebody who's been hypogonadal for 5 to 10 years, their brain is still responsive to estradiol with respect to 5-HT2A receptors, that you basically cause there to be this upregulation of these particular postsynaptic receptors. So if we're increasing synthesis, we're leaving serotonin in the synaptic cleft longer, and we've got more postsynaptic receptors, you would say that overall estradiol has pro-serotonergic neurotransmission effects, okay? So that's kind of a good thing, particularly since serotonin is important for things like mood, mood regulation, but it also plays a role in working and learning and memory. It plays a role in sleep, appetite, sexuality. So, I mean, again, serotonin is not just limited to, depression and anxiety. Now, let's add stress on top of this. Now, the two data sets that I'm going to talk about come from non-human primate research, where basically the animal was either a subordinate monkey or a dominant monkey, and they were studied later in life. And I think, as I said, I can't remember where I said this before, but I always say, if I had to be reincarnated, I would not want to be reincarnated as a subordinate monkey. It is not a good life. You have all kinds of health conditions. I mean, they've shown HPA axis dysregulation in these animals. They've shown immune dysregulation in these animals. And quite frankly, you can't muck around with the HPA and not eventually muck around with the immune system. I mean, they just work together, okay? And so bottom line is that it's not a good situation to be in. So what they showed is that as these monkeys got older, they looked at the effects of estradiol, either loss of estradiol or the exposure to estradiol. And the 5-HT1A receptor binding was altered in the subordinate monkeys versus dominant. And the addition of estradiol back was helpful, but it wasn't as sufficient in the subordinate monkey. And the same was true for tryptophan hydroxylase gene expression. They do have a response. The subordinate monkey does get a benefit of estrogen for TPH2, but it's not as robust as in the dominant monkey. So this is suggestive that there's chronic early life stress interaction with how estradiol impacts serotonergic functioning, such that you could set up the organism to be less, receiving less benefit from estrogen if they had early life stress. Okay, so that would be important when we get to some of the menopause research. So why study pregnancy? Well, I don't have to tell you that this is a unique hormonal milieu. So how many of you knew that during pregnancy that your glucocorticoid levels are sustained at a very, very high level? Okay. How many of you also knew the CRH was also consistently elevated? Okay. So the bottom line is that a few of you know, Teresa, of course, but the bottom line is that that is really like a crazy situation. That is not how our HPA axis usually functions, right? So for 9 months, you've got high levels of glucocorticoids as well as CRH. And we know that CRH and timing of CRH increases during pregnancy have been associated with things like timing of delivery. I mean, so my gosh, this is like a hugely important system to understand. And if a person's undergone a lot of early life stress, what impact could that have on their risk for maybe earlier pregnancy, earlier delivery, for example, or other adverse outcomes? The other thing I like to point out, and it's one of the reasons why brexanolone might actually be a good treatment for postpartum depression, is the fact that during pregnancy, you've got so much progesterone on board, and that progesterone is getting converted like crazy to allopregnanolone. Now, how many people think about allopregnanolone on a daily basis? I do. How many people have even heard? I mean, most of you have probably heard of allopregnanolone, right? Most? Okay. All right. Well, as you know, allopregnanolone is a very potent GABA A receptor agonist. And what do I mean by that? It actually works in the brain like a barbiturate and benzodiazepine. So not only does it increase the duration of the GABA chloride channel opening, but it increases the frequency of its opening in such a way that, quite frankly, if our brains were not incredibly flexible, we would be completely sedated our entire pregnancy. We wouldn't be able to get out of bed. So our brains have to be super flexible. But that's also one of the reasons why we think that for some people that flexibility doesn't quite work. They make the shift in the flexibility, but then after they deliver the placenta and the allopregnanolone goes away, that they wind up having these tremendous mood symptoms. And a lot of times those mood symptoms are not just dysphoria, but tremendous agitation and anxiety. A lot of like what you would see if you withdrew somebody from a benzodiazepine who had been taking a really high dose for 9 months, okay? And we also, like I said, know that these have effects on maternal fetal health and other factors. So I'm going to try and go through this pretty quickly because this is stuff you probably already know. But Kim Yonkers did a study where she followed women through pregnancy, measured childhood adversity, and looked at impact on birth weight and gestational age at delivery. No surprise. If you've undergone a lot of early life stress, you don't have always the best health behaviors. You might be smoking during pregnancy. You might just not be as healthy in general, may not be getting as much prenatal care. But even if you control for those things, you will still see an impact of adverse childhood experiences on time of birth weight and timing of delivery. We know that also if you've been a maltreated mom, that you're more likely to have harsh behaviors with your child, and your child is more likely to be maltreated. And so again, it's not all about the biology, so to speak. It's also that mother-child interaction, which is incredibly important. So basically, we've used our P50 to study multiple time points across this, lifespan for the female, primarily pregnancy and perimenopausal. And so I'm not going to jump right now into why did we study this group and what did we do. So we basically recruited a bunch of women, 1,500. I'm not going to go over those data today because we're still analyzing them. We're just finishing up, cleaning up the infant data. But we're basically had a hypothesis that childhood adversity increases risk of earlier birth, but particularly spontaneous preterm birth. Not just preterm birth. It's very important to think about the difference between spontaneous and medically induced preterm birth, because again, we have very, very few biomarkers of risk for spontaneous preterm birth. And that's one of the reasons we wanted to study adverse childhood experiences and whether that increases risk, similar to what Kim was showing. We also wanted to look at prevalence of birth outcomes. We really use that 1,500 as a way to recruit women for the latter part of this study. A lot of people were already looking at this question of childhood diversity and preterm birth. So we added that as an aim, mainly as more sort of to replicate some of the stuff that other people have been doing. But because we had to recruit 1,500 women, it gave us the capacity to get some of those women into our longitudinal study. So I didn't want to recruit them and get no information. So in any case, I don't think that's the most interesting part of the project. The part of the project that was, I think, the coolest is really the longitudinal aspect. So we've basically assessed these women through pregnancy. We did a bunch of psychophysiology. We also measured fetal adrenal volume. And then we looked at mom's and infant stress response at 6 weeks postpartum, or 6 months postpartum. And it's this latter part of the study that I'm going to talk about here. So again, we use the Adverse Childhood Experiences Questionnaire from the CDC Kaiser Permanente study. As you know, it has a rating of 10 items, 3 items related to abuse, 2 related to neglect, and then 5 types of household dysfunction. Now, I am not standing up here to say that this is as good as a child trauma questionnaire, nor am I saying that this is, completely comprehensive when it comes to adversity. It doesn't do anything about and talk anything about what's going on in the community outside your door. It doesn't say anything about whether you've got social supports from grandparents or what have you. But the reason we chose it is, again, we were trying to think about, okay, what is a kind of more sort of epidemiologically relevant. And these were studies that use this particular instrument at state levels. I mean, since the CDC study, gosh, there's been like multiple states that have confirmed that basically using this 10-item instrument, you get a sense of who's at risk. Who's at risk for poor health outcomes, whether it be cardiovascular disease, metabolic disorders, substance abuse, smoking, alcohol, other medical conditions. Again, that's the kind of mentality that we were going at this with, because we also wanted it, once we found something of interest, we wanted it to be something that would be easy for OBGYNs, for example, to put into their practice in a way that then can inform them about who in their practice is at risk. And so again, that's a long-winded way of saying we recognize it's not perfect, but this is why we chose it. We also were very interested in, again, in that timing question. Is a stressor or adversity that has its onset when a young female is hypogonadal due to being prepubertal, does that have a different effect on the brain than when she's postpubertal and she might be having these stressors that are occurring in the context of these hormonal fluctuations that would occur once she starts getting her period? So we basically sort of looked at pre versus postpubertal onset. Now, this is our initial data, and we're in the process of adding the rest of the sample. But, and this is one of those cases where, dear God, I hope that once we add the last 50 ladies to this study that we don't mess up our finding. But, all right, that's science, so you have to accept what you get. But in any case, I think we did a pretty good job matching our high ACE. So we said high ACE was women who had 2 or more adverse childhood experiences, and low ACE was those women who had 0 or 1. And we can talk about why we use the cutoff, but basically we did a sensitivity analysis in our menopause sample, and 2 was definitely a worthy cutoff. It also allows us to be able to use the entire sample instead of having to say only 0 or 3 or more, because then you've got a bunch of people that you would not be able to actually study and look at relative risk. So in any case, we basically showed that they were pretty similar, which is what we wanted, because we didn't want this to be about your current socioeconomic status. We didn't want this outcome about pregnancy, because we know maternal education has major impact on a number of different outcomes of pregnancy and fetal health. And so we wanted to make sure that they were somewhat similar. We didn't quite get it when it came to education. They were significantly different, but only in the sense that our high ACE women were less likely to have a graduate degree. So it wasn't like we were looking at women with just high school education in the high ACE group and then, college, graduates in the low ACE group. So again, I feel still pretty good that, there's not a huge amount of education differences here. So we took these moms, and at 2 times in pregnancy, we did 3D ultrasound and measured fetal adrenal volume, because we wanted to know, does mom's early life stress contribute to how her baby's HPA axis develops? And could that be possible mechanism for some of these behavioral outcomes that we see in the offspring? Could it also have something to do with how quickly the baby is delivered, timing of gestation, what have you? And so Eileen Wong's the OBGYN, she trained our research coordinator, and the two of them did ratings of all of these fetal adrenals, basically measured baby weight using ultrasound estimates. And so we got a fetal adrenal adjusted for weight of the fetus. And we also published that basically they did pretty well. The inter-rater reliability with respect to these measures was pretty good. And what we found is a maternal ACE by sex, fetal sex, interaction for adrenal volume. And again, it's weight adjusted adrenal volume, because males would typically be a larger baby. And so what we're seeing here is, if you look in blue, you can see the difference between the high ACE males, I'm going to call them high ACE, but baby boys of high ACE or low ACE moms, and baby girls of high and low ACE moms. And what we can see here is that the effect is in the opposite direction for either sex, although I would say that we're not really seeing a huge effect in the girls of mom's ACE, and that this effect is really being driven by the effect of mom's ACE on boys. Now, we can talk till we're blue in the face about this, but there is quite a bit of evidence in animal studies and a growing evidence in human studies that what happens during pregnancy with respect to maternal stress does seem to have more of an effect on outcomes for males than for females. And so this would also suggest that even though these women, and I didn't say this, their perceived stress scale scores were very similar, whether they were high or low ACE. So it wasn't that the high ACE women were more stressed during pregnancy. So we might be also seeing here that even early maternal adversity has a more of an impact on the male offspring, at least with respect to this particular marker, adrenal development. So then we took those babies, and when they got to be 6 months of age, we made another addition to the study where basically we were going to assess their HPA function at 6 months postpartum. So we brought them in, and we basically took them from mom. Well, we did salivary cortisol on the baby first, got a couple of baselines while the baby was there relaxing with mom, then took the baby away, put it in a car seat in another room, held its arms down for 3 minutes and didn't look at the baby. And we then blew a bullhorn, like you have at, games. And we blew the bullhorn 3 times. And then so that was our stressor. And we got that past IRB. And the bottom line is that we had some babies that actually acted like it was no big deal. Again, talking about baby temperament. Babies are obviously born with a different temperament. Some babies definitely cried, and then other babies just got mad. So the only time we had to abort this particular stressor is if the baby cried for at least a minute straight. And then we basically aborted the session, did the salivary cortisols and all of that, and took the baby back to mom. So our research coordinators were in our meeting. We were all together with our basic science colleagues. And so Jess and Grace basically said, in doing this study, we can tell who's a high ACE mom and who's a low ACE mom. And we were like, that's kind of interesting. Again, you have to understand we were there with our basic science colleagues. We were all talking about this together. And they said, well, yeah. And we said, well, what do you see? And they said, well, our high ACE moms don't ask a lot of questions. And our low ACE moms really, they're asking us, please bring my baby back. If you promise, if she starts crying, you're going to bring her back. You know, they just showed a lot more concern. They were, you know, a little anxious about it. And they expressed more, you know, sort of agitation about this particular process. And so we were like, wow, never thought that this would be stressful to the parent. And then we said, well, maybe we should actually measure mom's stress response. And then we were like, okay, we'll do that. And then all of a sudden, Tracy and her postdoc said, you know what, we can do that in an animal. So let's make an animal model out of that. And we were like, okay, all right, well, this would be a really cool translational study. So this is what we did. We started then adding moms to the picture. So this is the paradigm, and we were able to publish this in Biological Psychiatry, including both human and animal data, which is a really cool thing to be able to do. So mom and baby separated, undergo various serial cortisol assessments, and this is mom. So basically, the dark boxes represent the cortisol response for the high ACE women. So not only behaviorally were they more chill about this separation, they actually didn't have a physiologic response of stress. You know, it was dampened. I mean, it had a little bit of a response, but you know, it's not like huge peak there. The low ACE moms had a lot more sort of not only behavioral, but also a physiologic response of stress due to this separation. Okay. Now let's look at the animals. So Katie made a box where she took mothers who had had pups. Now, either the mother was a control mother, or in their paradigm, to try and mimic the ACE score for a human, they use a chronic variable stress model. So they didn't just do one kind of stress. They tried to create more of a stressful environment for the mouse. And they did these stressors, what we call peripubertally, because they were after weaning. But you know, the mouse lifespan is really short. So if you do 2 weeks of a stressor and there was a different stress every day, that's the chronic variable stress model. And so you wind up getting into that sort of peripubertal window for the mouse. So either you were a stressed mouse or you were not a stressed mouse, and you could be in a box with your offspring on the other side of a permeable barrier or semi-permeable. You could smell, you could hear, you could know that your mice pups were on the other side. And so what we measured is how crazy the mom did. How much movement was she having on that side of the box trying to get to her offspring? Because the natural thing to do for the mouse mom is to try and get to her offspring, you know, to bring them back to the nest, okay? So this was a stressor, again, a maternal separation. And what we found here is that in the animals who were controls, basically as they were going around and moving around the cage and demonstrating sort of agitation about not being able to get to their pups, they also had an increase in corticosterone that was correlated with how much they kind of went after getting their mouse pups. And then if you look at the stressed, you can see that is an opposite direction, that they not only didn't work as hard and didn't travel as much around the cage to get to the mice, but there was less of a relationship with the corticosterone. And then if you look overall, you see exactly what you saw in the humans. You see a dampened HPA corticosterone response in those peripubertally stressed mouse moms. Very cool, right? Now, the thing that's really cool is you can actually go in and take out the periventricular nucleus of the hypothalamus of the mouse. You can't do that in a human, obviously, and obviously do things like RNA-seq and all these really wonderful sort of new age, laboratory methods to say, okay, what's happening different? And basically, in the peripubertally stressed mice, there was about 24 immediate early genes that were upregulated in that mouse versus the control. So suggesting that HPA axis control over the stress response in that peripubertally stressed mom is different, and that these genetic differences could lead them to having a more dampened stress response. So what about the infants? So Mary Samuel is my dear biostatistician colleague, and we basically looked at high ACE versus low ACE moms and their babies. And again, what did we find? We found that at 6 months postpartum, there was a maternal ACE by infant sex interaction with respect to glucocorticoid response to the stressor, okay? Such that here you see an effect in both males and females, but the effect again being more predominant in the males. But again, it's not so much that the effect is in one sex, it's the fact that the direction of the effect was different based upon infant sex. So one of the things that I deduce from these data, both the fetal adrenal volume and the baby's HPA axis response, is that you have to control for fetal sex in order to see the effect of mom's ACE, okay? And if we just wanted to look at difference between boys and girls, we wouldn't see an effect if we didn't control for mom's ACE. So I have to say, anything my career will possibly do, I think that hopefully the body of my work over the past 20 years will be that everybody should be really thinking about the reproductive stage when they do research, particularly if they're looking at females, because these hormones have dramatic effects. And two, sex matters. The sex, if you're going to study males and females, you really do need to be thinking about potential sex differences, even if you don't think there's going to be a reason for one to be there. I still say always look at sex. And then we never, hardly ever in our clinical trials or imaging studies actually look at people who've had early life stress versus those that don't, unless you're interested in that topic. But say, for example, if you look at, say some of the drug studies that we have as far as antidepressants, there was one study that sometime past basically showed that it was only in people who had had significant early life adversity that they responded to this treatment. And if they hadn't actually looked at that and hadn't looked at something like a peripheral marker, like a cytokine or something like that in relationship to that early life stress, they wouldn't have found the effect of the treatment. We also know that a lot of disorders, if you've got major depression or bipolar disorder, and you have early life stress and adversity or trauma, that you don't respond as well to the treatments typically. And that's something that people see clinically, and there have been some reports of that in the literature as well. So I think, again, we have to be thinking very much about early life stress as a potential factor that can add heterogeneity in our outcomes. So just to very quickly summarize the peripubertal piece, and then I'll talk about the menopause quickly, and then we'll get to Q&A. So most ACEs in this particular study occurred in the prepubertal window, which I didn't talk about before, but just to suffice that at least in our sample, it seems like when people have two or more adversities, they really do occur very, very early. And again, you can say, okay, well, let's think about what brain structures may be developing during those times. Preclinical model does mirror the same thing that we saw in the human of a dampened HPA axis response in those that were stressed in childhood. Again, consideration of sex is really critical. And the question that you might want to have and discuss in the Q&A is that, again, the data suggests that in the peripubertal window, particularly prenatal exposures, that males may be more sensitive to females to the effects of maternal stress. Whether, again, this is true for, moms who weren't particularly stressed during pregnancy but had early life stress, these data would suggest that that is the case. And then dampening of a stress response. If you're going to be born into a very stressful environment, then it may actually be a good thing to have a somewhat dampened stress response. But then if you think about it, over decades and decades and decades of having a dampened stress response, that's probably not good for your immune system. So are we setting people up to say that this dampened stress response, at least initially, is a good thing, and that's adaptive? But over the long haul, maybe that's not so adaptive when it comes to long-term health. And again, that's only if your HPA axis response to the environment is actually matches the needs of the environment. Because obviously, if you're raised in a very stressful environment, you don't want to be having your HPA axis getting, revved up every single day, all constantly. That's not a good situation either. So again, what's adaptive and what's maladaptive is a real question. Okay, so moving on, why study menopause? Does anybody have a good thought about that? Well, we live a long time in the postmenopausal state. And I think I said this at the retreat, that let's think about it. If we died You know, in 1918, the average life expectancy was about 55. Average age of menopause in the United States today is 52. So we'd be dead, you know? So the whole concept of menopause is actually new to our species. Our brains do not develop that quickly. And as one of the gynecologists used to, she said to me when I was talking to her about a patient who I wanted to have her on estrogen, and I go, you know, this woman's really healthy. She's only 60 years old and she's going to live at least another 20 to 30 years. And the gynecologist said to me, well, yeah, I know. God gave us the capacity to live longer, but he didn't tell us it was going to be fun. I was so infuriated. I was like, what kind of women's health advocate are you? I mean, geez. I mean, like, I don't know. I was like, you know what? I think we can do a little bit better than that. So what we really have to do is try to understand who really needs estradiol in order to be able to have a healthy brain, healthy mood, sleep well, not have a lot of anxieties, not become psychotic. I mean, seriously, I mean, there is another peak of psychosis onset in the peripubertal window. I know you ladies, you guys have a lot to look forward to, but all I could tell you is that I know, so I get referrals quite a bit from people saying, oh my God, this mother has now become paranoid. I mean, she's really paranoid. And I'm like, well, yeah, she's probably perimenopausal and we need to do something about that. And so I have saved a few people from frank psychosis by treating them with estrogen. Okay. So all I can tell you is that this stuff is real. All right. There's not a single body tissue that isn't affected in some way, shape, or form by estrogen. You know, just the epithelial cells that line your body. Oh my God, it could go on and on and on. But we're going to talk about cognition and mood. So I was able, when I went to Penn, to join up with Mary and Ellen on the Penn Ovarian Aging Study. This study had already been going on. These women were recruited in the greater Philadelphia area by, guess what mechanism? Random digit dialing. Since when have you ever thought that you could recruit 400 and something women just by calling on the phone their landline? Their landline. I know we had landlines in 1990s. I mean, it's like blows me away. But in any case, 50% of the group was African American, 50% Caucasian. This is representative of the Philadelphia region. And also, we just did not have a lot of data in the 1990s about sort of race and how that might interact with hormonal changes across the menopause transition. And so they were very curious and wanted to sort of really investigate this a little bit more. One of the things that was really important is that everybody was recruited as a premenopausal woman. So they all had regular menstrual cycles. And so we were able to get data from even the most earliest changes in hormones and how that could affect mood, cognition, sexuality, onset of vasomotor symptoms, onset of sleep issues. I mean, they basically looked at everything. These women were studied yearly. With hormone levels, cognitive assessments. Now, Ellen and Mary were not neuropsychologists, so they didn't do a whole huge battery of cognitive assessments, but they did include processing speed tasks because we know that age affects processing speed. And there had been some data at the time that estrogen level impacted your verbal memory. And so they had two domains that they tested, processing speed and verbal memory. And basically we went all the way from premenopause to postmenopause, which is being without a menstrual cycle for 12 months. And this slide shows the risk for depression or the prevalence of depression across this whole cohort. So if you start over here where you see the arrow, that represents the final menstrual period. So they looked at proportion of women who had depression in the years leading up to the final menstrual period and the years after the final menstrual period. The light blue bars represent women who had had a pre-menopause depression. So they were either came into the study with a history of depression, or they had a depressive episode that was observed in the study before they became perimenopausal. The dark blue bars are the women who had depression only in the perimenopause. And what you can see is that the prevalence decreases some in the group that have had a relapse, but that basically in those dark blue bars, those are women who only get depressed during these hormonal fluctuations. So one of the questions that we had is based upon the animal studies of estrogen effects on serotonergic functioning, could these women here be women who have more adverse childhood experiences? So we went back and looked at the timing of depression in relationship to puberty, in relationship to perimenopause, as well as whether you had onset of your ACEs in the prepubertal window versus the postpubertal window, because we were really interested again in this timing issue. And just for the sake of time, I'm just going to summarize what's here. Bottom line is that if you had one ACE in the prepubertal window, which is this light bar, you were at decreased risk for depression, either across your lifespan or in the perimenopause, even if you went on to have more ACEs in the postpubertal window. So there's some suggestion here that one adversity really early in life may have had some stress inoculation effects on the offspring. On the individual, sorry. Suggesting that Nietzsche was right. What doesn't kill you makes you stronger. But then we wanted to also know what's the effect of ACE on cognitive aging. And that basically menopause, does it have a sort of a independent effect on cognitive aging? And does that interact with ACE? So he first had to show that menopause had an independent effect. That had never been shown before. Most people who had done longitudinal studies of what was happening across the menopause transition basically said, well, women are just getting older, and everybody as they gets older, they have worse cognitive functioning. But this was the first time that we were able to show, because we had about 3,000 observations on all those individuals. And we had a lot of repeats. A lot of people stayed in this study. And we knew when they were going through each phase of these hormonal changes, because we got menstrual cycle data as well as hormone data. We measure perceived stress, depression, anxiety, BMI, education, performance at baseline. And we still showed that with immediate recall, if you look over here, you can see the premenopause baseline, that by the time they became postmenopausal, there was a significant decline in their ability to recall a list of words. This was just a list of 16 words. The delayed recall showed up as early as the early transition. So this is when women are not even sure that they're in menopause yet. So what I try and tell when I go to OBGYNs and give this talk, I say, do not poo-poo your woman coming into the office and saying that her memory's not as good. Don't tell her it's just about aging. And you might be able to help her with a little bit of estrogen. So don't discount that. And that was something that was really novel at the time when we showed this, because again, nobody had ever been able to say that there was an independent effect of estrogen, or of menopause. So again, we looked at the serotonin estrogen interaction, because we want to know now, does early life stress impact that cognitive decline? Because one of the things that we looked at in that sample, we were able to see that, yeah, some women actually did pretty well across the 14 years of the study, and they either got better at the task or they had only a slight decline. And we were really curious about those people who had more of a decline, and whether there was an adversity impact on how women's brains respond to being hypogonadal. So we did something called the tryptophan depletion paradigm. So the tryptophan depletion paradigm, how many people know this paradigm? Okay. So basically, you give people a bunch of amino acids. So we give them 72 capsules of amino acids, and these are big capsules, and they have to take these within 20 minutes on the morning of their tryptophan depletion. So they undergo these procedures 4 times. In the hypogonadal state, they came in and they did an active depletion or a sham depletion. Then they were randomized to estradiol or placebo and underwent the sham and active depletion again. So everybody had 4 sessions. Now, in the hypogonadal state, after the tryptophan depletion, at each day they would undergo brain imaging and cognitive testing. And then after either the estrogen or the placebo, the same. They would undergo brain imaging at the end of the tryptophan depletion. It only takes about 6 hours to lower blood levels of tryptophan by about 80 to 90%. And there's studies that show that this also results in a dramatic decrease in serotonin in the brain. Okay, so it's a complex study paradigm, and it was very difficult to get the sample, but we did. And we used something called the NBAC. This is a working memory task. It robustly activates the DLPFC and the middle frontal gyrus, part of the executive network. And it particularly, as you get more and more difficult in the task, you get more robust activation in these brain regions. So study population, 40 to 60 years of age, within 10 years of final menstrual period, 60 enrolled, 33 completed. Outcomes, we're focusing on working memory. So just for the sake of time. So we basically did a whole brain analysis and looked at an interaction. So where did you see in the brain an interaction while the people were doing this task? And this is the 0 to 3 comparison, the 0 back to 3 back comparison. Where did we see basically an interaction between tryptophan depletion and ACE? Because half the sample had high ACE, half the sample had low ACE. And this was, thank God, we saw a robust interaction in the LPFC. Thank goodness. All of you that do brain imaging know that you're like, yeah, at least it's in a task active region. Interestingly, we do have some insula findings as well, but we're going to focus on the DLPFC. So basically, as you can see that as people's performance improves, they have an increase in bold signal in the DLPFC, because you basically now extract that signal and you can do all these statistical analyses. We also showed that even though there wasn't a huge difference in performance, I mean, the high ACE people did a little bit, they were a little bit slower in getting the true positives. They got the same number of true positives, but they were just a little bit slower in their reaction time. Interestingly enough, the higher the ACE, the more you have to activate the DLPFC. So could this be an example of, okay, if I've had a lot of early life adversity, in order to do as well on this task, I need to activate this brain region more. Again, a lot of people do a lot of hand waving when they interpret their functional imaging data. Hopefully this doesn't sound too much like hand waving. All right, so let's see what difference we have between the low and the high ACE people when they underwent tryptophan depletion. So right now, the white bars are sham depletion. So I don't do anything to their serotonin levels. Look at the baseline difference between high and low ACE people. And then if I tryptophan deplete them, the impact of changing the serotonin level has an opposite effect from baseline or from control in the high versus low ACE ladies. And I just want to give Sheila, Sheila was my MD PhD student in this particular project, and James is my imaging colleague who basically gets me through all of this. So basically, this is what we saw here at baseline. This is when the hypogonadal state. This is what happens when we give people placebo, nothing really significant. Look what happens when we give estradiol. Basically, the high ACE ladies and the low ACE ladies look exactly the same. So what does that say to me? Well, as somebody who tries to figure out who should take estrogen, because there are risks of taking estrogen, but maybe people who have high ACEs, really, their brain may really need estrogen more than people with low ACEs. And so is this of clinical relevance? That's a really good question. The other question I have is that maybe it's adaptive, that extra activation in the high ACE group, maybe that's how they actually perform as well. And so I'm changing their adaptation. Well, I would actually think if, I mean, I could get into whether I think this is a good thing or a bad thing. Overall, I think it's a good thing to make their brains look a little bit more like the low ACE ladies. But again, we can argue about that a little bit in the Q&A. Very quickly, we also saw an effect of ACE on network connectivity. Within network connectivity is associated with better performance on executive functioning tasks. And what we see here is that within network connectivity declines among people with higher ACEs. And that's actually not thought to be a good thing when it comes to executive functioning. So ACEs contribute to menopause-associated risk for depression and cognitive declines. Estradiol differentially impacts neural activation in high and low ACE women. And overall, the research presented here suggests that there's alterations in how the brain of the older woman who is hypogonadal, what her brain does when she's given a working memory task, if she's had ACEs in childhood or not, and then how her brain responds to estradiol. I don't have time to talk about our inflammatory studies because we've also looked at cytokine profiles across the menopause transition. And we've also looked at the impact of ACE and affective symptoms on cognition across the menopause. And we've got a bunch of other studies that I can't possibly go into at this moment. But in any case, have a lot of people that done a lot of work to help us get these data. And then we obviously couldn't do this without our funders. So I'm going to stop there. I'm going to thank you for your attention. And if you have questions, I'd be happy to address them, and hopefully in an adequate way. Thanks. Okay. Yes, Christina. Yeah, yeah, yeah. Well, I would love to have had a large enough sample to actually also look differentially at weight, but you know, the truth is that, I mean, weight was, I mean, in the larger sample where we had the 3,000 observations, BMI was correlated with cognitive performance. So that's that's why we included it in the model, because some portion was involved, you know, BMI, the higher the BMI, the worse the cognitive performance. So we did actually see that. Did I look at that as my primary, effect? No, but we included it because we expected to see it. So yeah, but as far as how the brain changes, I don't think I have a large enough sample to actually look at BMI. Could be interesting though for a future study. Other question? There was another question. Yeah. Oh, here. Bob, the person right behind you first. Let him go first. Oh, what? You're Russ? Not Russ. No, God, I'm blanking on your name. Tell me your name again. Okay, Corey. All right. What's up? Well, there are a lot. Dopamine, for one, is quite affected, particularly in the DLPFC. Dopamine has very potent effects on COMT activity. And so, depending upon whether you've got very robust COMT, it basically decreases COMT activity. So, depending on whether you're a Val-Met, or Met for the COMT 156, whatever, you know what I'm saying. It's basically depends on what your baseline uh, dopamine function is in the PFC, whether probably having high levels of estradiol helps you with respect to executive functioning or worsens your executive functioning. We do have COMT genotype on our ladies, but we've not looked at it in relationship to the outcome, mainly because, again, doing genotyping in 33 individuals is limited. And so, but yeah, there are many other, I mean, acetylcholine is affected, norepinephrine is affected, glutamate is affected by estradiol. We have some data using spectroscopy to show that executive functioning in postmenopausal women is associated with the level of glutamate in the DLPFC. Having higher levels of glutamate in the DLPFC, you could have that for many different reasons, but when we give a psychostimulant and we increase dopamine in the DLPFC, we drive down glutamate and they actually perform better. So, and there's some relationship between how much we drive down glutamate in the DLPFC and their performance. So that's another area that we're looking at this point as well. Mm-hmm. Bob. Oh, yes. Uh-huh. You know, if I could find a good peripubertal researcher who wanted to come, if we know of somebody who that's really what they want to study, I would love to add it. I have not collected that cohort myself. It's just a cohort that I've never focused on as far as actual data collection. So it's only that I can go back and ask the people that are now 50 to 60 years old about what happened to them at puberty, but it's not that I've collected a pubertal sample myself. Mm-hmm. No, I don't disagree. It's a really great population. Oh, absolutely. Yes. Yeah. No, absolutely. I just don't have any money to study them. And not being a child and adolescent psychiatrist, our clinic was for adult females. And so the youngest that I typically saw people were 16, 17. And that was relatively rare that I saw 16 and 17-year-olds. Yeah, Mark. Mm-hmm. Mm-hmm. Well, that's a good question. I mean, I think that it's confounded, particularly menarche. Menarche is often confounded by a lot of different societal, not, I don't know that people have looked specifically at ACE. I don't know. Aviva, do you know if people looked at specifically at ACE and age of puberty? But I mean, obviously, being raised in a more stressful environment, I've seen for various reasons, whether it's, you know, poverty or, um, you know, the community violence, those kinds of things. My impression of those data is that they have earlier onset of puberty. There is, but I don't think that that explains the stress piece, and the community adversity, where you're raised, what kind of stressors are you dealing with in society. But I don't think I've seen anybody look at ACE score specifically in age of onset of puberty. Um, I think when we looked back at our data, age of onset of puberty was not different between our high and low ACE women. But again, that's reported now they're 50 and 60, and they're reporting when they had their menarche. So I'm pretty sure that we looked at that and there wasn't a difference. As far as menopause is concerned, there have been some data talking about even, say, for example, having a major depressive episode increases your earlier menopause or is associated with earlier menopause. Um, I'd have to look and see if our high ACE ladies differed in the penovarian aging cohort with respect to when they went through the stages. We control for when they went through the stages, but I don't know that I actually looked specifically to see if that was affected by ACE. But it's a good question. Mm-hmm. Yes, they all healthy. So that brings up an issue. These were relatively healthy women. These were women who had undergone anywhere from 2 to 6 ACEs, nobody above 6, because we couldn't get people who had 7 to 10. They were sick. They had symptoms. So no one had had a major depressive episode within at least a year. No one was taking an antidepressant. And the vast majority of them had never had any psychiatric symptoms to speak of, or at least that, a SCID interview picked up on. Didn't mean that their lives were not, I mean, it's interesting. The higher the ACE level, the less likely in some ways the women were to complete everything. I mean, it just, again, you could see the impact of having adversity. But, we did our best to try and control and make sure that people were as similar as possible currently, so that we could say, okay, how much of this is due to the adversity they experienced earlier and the enduring impact of that. So I should have made that clear that these were all very healthy ladies. So that increase in brain activation in the DLPFC during doing working memory, that's adaptive for them in this hypogonadal state. Now, see, what I don't know, because I didn't study them prior to becoming hypogonadal. So when does that increased activation occur? Because if they're the effect of estradiol at dampening that, it makes me think that when they were or at least reproductive aged, that they weren't having to activate like that in order to do as well on the working memory task, and that this really is about being hypogonadal. But you can always argue that, that's why we had to have the placebo, obviously, because is it an estrogen effect, or is it that this was just always there? I think that this is really an effect of being hypogonadal, particularly because of what the animal studies suggest. So I've really struggled with, if I give them estrogen, am I getting rid of their adaptation? And, adaptation isn't always pathological. I mean, people talk about the capacity to adapt as being incredibly adaptive. I mean, that's really good. So am I getting rid of their adaptation? And then it makes me think, well, what biologically am I looking at? So if we think that when you're stimulating pyramidal cells, for example, in DLPFC, you're releasing glutamate. I mean, that's really, people talk about the bold signal at least being partially this, neuronal glial glutamate phenomenon that then, basically every millimolar of glutamate that the glial cell picks up supposed to send off a, pseudopod and releasing this substance that causes vasodilatation in that particular brain region. And people have said, okay, well, that's partially what we're measuring, or at least seeing with the bold signal. So, because you're getting this increased blood flow, but obviously there's oxygenation differences and those kinds of things as well. But let's say if you're stimulating the nerve cell and you're releasing glutamate, if you have to release more glutamate to get the same effect of cognition, that might actually be okay acutely. But then if you do it chronically, and as we age, our glial cells age as well, and maybe your glial cells aren't able to soak up that glutamate quite as effectively, are you getting over decade and decade neurotoxicity due to perhaps seepage of this glutamate? So I just wonder. I don't know. I mean, that's where I'm wondering if the estrogen is really important, because if you can kind of dampen down the amount that they actually need to activate, then that might actually be a protective factor. So animal studies would be really helpful. Yes, Allie. Yes. Oh, you mean within the high ACE group? Within the high ACE group. So within looking, let's say if we just looked at these people here, I did not just look at that group. Yeah. That would be true, but also too, you would think about their performance. So at least, although that's only acutely. So basically, these women performed pretty similarly. This is not like studying people with schizophrenia versus healthy controls. We know that you're going to see huge differences in performance on an MBAC. These ladies, I mean, we're talking about very minor differences in performance. So that's why I have to say, well, this is adaptive. This, whatever I'm seeing here, is adaptive, um, but that doesn't mean that it's good for long-term health. Sort of like the babies having a blunted HPA axis response if your mom's high ACE. If we know that high ACE moms are maltreated or more likely to maltreat their or the child, even if it's not them, it might be a partner who maltreats the child, or you might be, in a more stressful environment just in general. So maybe not having a glucocorticoid response to every single little stressor is a good thing. But we also know blunted glucocorticoid response, particularly in males, is also associated with sociopathy. So that's actually not good either. um, so, okay. And then after decades, so maybe acutely it's good, but after decades, it actually could potentially alter inflammatory response, for example. And, the data from the risk study out of the UK shows that, again, adversity, even in victimization in childhood, is associated even only at 18 years of age. You can also already see cytokine differences. And so we just know that, these kinds of situations are not good over the long haul. Yeah, hi, Emily. Mm-hmm. Mm-hmm. Um, oh my gosh, I'm trying to remember. I'll have to look it up and get back to you as to whether they're, yeah, it's okay. I'm just trying to remember, like, why did we not report on that one as much? I think it just wasn't quite as interesting, but I'll have to go back and look and see what's there. But yeah. Hmm. Yeah. Yes, we did. We did a lot of perceived stress. So when they came in and did in the 1500, we did not follow them over the whole pregnancy. We really used the 1500 as a way to recruit the longitudinal sample. So all we have on the 1500 is they came in within, I think it was 8 weeks to 20 weeks, so or 17, something like that. And we measure perceived stress, Edinburgh Postnatal Depression Scale score, ACEs, and then got their consent for us to extract their infant record and extract their obstetrics records. So we have relatively little information on a large number of people. So we'll have to go back and look. But we do have at least their ACE scores and what have you. The other sample where they did the fetal adrenal volumes and we did the psychophysiology. So we have acoustic startle response on these ladies and salivary cortisol, both in pregnancy and in the postpartum period. So we were looking at arousal regulation because, again, thinking that if you've had early life stress, maybe you're hyper aroused. And is hyper arousal not just a representation of what's going on with HPA axis, but also sort of psychophysiology in general, whether it's sympathetic nervous system or what have you, and whether it's actually the arousal regulation that's associated with the earlier birth. So we haven't finished analyzing those data yet. So we have a lot of data. Uh-huh. Nope. Nope. I am not an attachment person. And believe it or not, at CHOP, Children's Hospital, I went to them and said, is somebody here, does somebody here know how to measure these things? Because I don't know how to measure them. I could not find a really good perinatal infant collaborator. I could basically, we did get developmental assessments on the infants. But as far as measuring mom infant stuff, I just didn't have a good collaborator on that. And so again, I had to kind of leave things off. Um, but I did look for a good collaborator. So it's one of the reasons I got really excited coming here with the Harris Infant Mental Health Program and DPRG. There's so many people here who know how to assess mother-infant interactions in a much more sophisticated way. So I always thought, well, if I were to do this study again, I would certainly add those kinds of measures to the work that we're doing. Yeah, hey, Laura. Yeah, it's a good question. You mean in the perinatal population? No, I did not do cognitive assessments in moms. And babies, we stopped following at 6 months of age. Oh, in the menopause, no, we do have a huge number of batteries. We have a lot of batteries. We have every potential domain assessed. And I did not do in this sample like a Brown Attention Deficit Disorder Scale or some kind of measure of executive functioning, but we did things like DSST, symbol copy, we did emotion identification, we did sustained attention, the CPT. So sustained attention might be a good measure for executive functioning. Obviously, uh, if Trails A and Trails B makes any, it's kind of a poor man's test. But did we do anything other cognitive? We didn't do like a Tower of London or anything like that. So I'm trying to think about, I thought we had a cognitive flexibility test, but they did all of that outside of the magnet. So we have tons more data to evaluate. Yes, Christy. Mm-hmm. Yeah, yeah. Yes. Boys, decrease for the girls. Yeah. Well, female offspring. So but at 6 months of age, you could argue that maybe it's an effect, that you're seeing an effect in females, because that's where we saw more of a difference between the high and low ACE baby girls. It still wasn't huge, and there was a big. I mean, those were standard deviation bars. So I mean, there's a lot of variability, and um, so yeah, I mean, it could be that, again, I'm not ruling out, at least in those data, that the girls are not being affected. And I mean, I think for one, it would be really great to follow up these infants, not this cohort, but to get a new cohort of infants, do some similar, I would leave some stuff out of the study. I'd probably add more stuff on attachment and those kinds of things to the study. But that would have to be somebody else taking that on. And, really try and follow these kids over a longer period of time, hopefully get them into school age to see, things like sustained attention, ability to, focus, cognitive flexibility. But then, of course, you've got all those measures that in the parent as well. So it becomes a much bigger cohort. And, but in any case, yeah, it could be also the girls are affected as well. Yes. Yes. Yes, they are. Yeah. Yeah. Well, it's a very good point because you start to wonder, you know, not all stressors affect people equally, and there's something about being a mother of a 6-month-old child that it might be more sort of an ecologically relevant stressor. This kind of maternal separation. It's one of the reasons why we use that model in the animal too, because they get very stressed out when they're separate. If we did like a Trier social stress test, which is a social evaluative stressor, all I can tell you is that we have done some of those in other samples. Practically every sample of women that we look at, they have a dampened stress response by ACE. And again, we're looking at people who don't have PTSD, people who don't have depression, any other SCID diagnosis, that adaptively, maybe that's a good thing that they have less of a stress response. So I don't know. But we keep seeing it over and over again. We went into this thinking, oh, the reason why early life stress is going to lead to earlier birth is what we thought we would see heightened psychophysiology, so heightened acoustic startle response, and we thought we would see heightened cortisol response because we thought, okay, this is going to be like you get increase in CRH, you get increase in cortisol, and that's going to push you to have a earlier birth. And that's just not what we were seeing. I mean, at least even in our pregnant sample, we did one study where we look at microbiome in pregnancy and stress response. And we definitely still saw in the high ACE group a dampened stress response, even while they were pregnant. So, okay, well, we'll see at least we're consistent. All right, it is over, it's time. Everybody, I'm sure, has other things to get to. And I thank you very much. It was a pleasure to be here today. All right.